So this I have actually taken from online only. It's from McGraw-Hill, as you can see. So what are we going to see over here? It's going to be about the different kinds of cells that are present, what exactly are cells, and how are they responsible for the structure and the function of any kind of living organism. We must have learned as when we were young that the cell is the basic unit, and the structure, like basic structural and functional unit of life. So this is where we are going to see, and how is it important for our body, and what exactly is going to happen when there's any kind of abnormality, and what exactly are we going to do in case of therapy. So this is one thing, and then who had discovered the cells, the basic history as such, the different kinds of shapes. And I found this particular PowerPoint a little better. There are others also that I have basically all taken from the internet. So at any point of time, I can just share it with you if you want. So we'll just, have a look at it. So when it comes to the basic cell theory, the study of cells is cytology. And Robert Hooke was the first scientist, in fact. He had coined the term cellulae. So he had actually seen, what do you call it, the onion cells. He had taken the onion peel and studied it under the microscope. Theodore Schwann is one person. He said that, all the cells, all the animals are made up of cells. Louis Pasteur, he had demonstrated that cells come from pre-existing cells. So this has, just a second, I'm sorry. Read a book. So when it comes to the cell theory, it's that all the organisms are made up of cells. It is the basic structure and functional unit of life. All the structure and the functions are because, every, as such, cells consist of, you have the tissue is consist of cells, tissues make up organs, organs make up organ systems. So if one cell doesn't work, it means that your organ system is going to fail at a particular point of time. So, and then when it comes to the cells, any cell has to come from the previous cell itself. That is your cell division. You'll come to know about it in the later chapters. And then you can see that a particular cell in a tissue will have same kind of properties. For example, your pancreatic cells are going to give the different kinds of hormones, your insulin, what do you call it, your different kinds of hormones as such. So they are not going to give you something else, like, they are not going to produce thyroid, thyroid hormone if they are from the pancreas. So they have basic similarities. And what are the different kinds of cell shapes and sizes that are present inside the body? You can get to see that more than 200 types of cells are there. You have squamous, which is like thin, flat. All this is basic, your intermediate, which we are going to go into much more detail. Squamous, cuboidal, columnar, polygonal, etc. So different kinds, again, you can see these are the squamous cells. They form the lining, the squamous epithelium as it is known as. The epithelial cells are again squamous, cuboidal, columnar, polygonal. In this manner, you have different kinds of cells, and based on the presence of where they are exactly present as such, in which kind of tissue they are present, they have different names. So squamous cells form the lining, cuboidal cells are present. Then this columnar cells, they have ciliated epithelium on the top. Polygonal cells are of the shape. Stellate have mostly, what do you call it, they are present, in the nerves. And spheroidal, discoid cells are seen in the RBCs. Fusiform cells or spindle-shaped cells, these are present in your muscles, and fibrous cells. So when it comes to the size, we are going to talk about very small. They are not visible to our naked eye. Most cells are around 10 to 15 micrometers in diameter, and you get to see that some of the egg cells, they are 100 micrometers in diameter. This is the basic length that we are talking about. And nerve cells are more than a meter long. But how are they present inside the body? They are connected end to end. So some nerve cells, only one nerve cell, if you can see, it can be a more than a meter long, but condensed. Myelinated, non-myelinated, again, that is a different topic altogether. But if they are covered, they're like, with the myelin sheath, they are known as myelinated. Otherwise, it is non-myelinated. And there is a particular cap. That means the cell size cannot be more. If a cell becomes big, you have a term for it, which is like, hypertrophy. So the cell cannot become large. Then it can lead to a disease, tumors, example. And then what will happen? It will burst. So pus cells, example. So any increase in the diameter and the volume is also going to increase, and because of which the surface area will increase, because of which it is going to cause a problem as such. So this is the cell shape and size. If you see small cells and large cells, they're just given the basic rough, the surface area and the volume as such. And what are the basic components of the cell? How are you going to view a cell in the first place? Microscopes. So you want to see a simple microscope, compound microscope, phase microscopes. You have, and if you want to see certain bacteria and all that, you're going to use phase contrast microscope, wherein, dark background, you're going to see the, like the microorganisms or the cells as such. Otherwise, when you want to see in the best and the finest way, you're going to see it in the SEM and the TEM. Our CCMB has these like microscopes, the transmission electron microscope and the scanning electron microscope, wherein what happens is, in the transmission electron microscopes and in the scanning electron microscopes, you can see the finest of the microorganisms that are present and finest of the cells that are present. So what are the basic components of a cell? If you see again, is there any cell that is visible to the naked eye? That's the human egg, which is 100 micrometers in size. So you can actually see that, like when what do you call it? When when you do a scanning, a basic scanning of the abdomen and the uterus, you can actually see, if it is the ovulation time, you're going to see the human egg attached to the lining of the uterus and the fallopian tubes. So it is visible to the naked eye. And in the light microscope, with a resolution of around 200 nanometers, micro nano, 10 to the power of minus 6, 10 to the power of minus 9. So over here, you're going to see the basic human cells. You can see the different kinds of faces also. If it is a stained, like, slide that is available, you will be able to see the different kinds of cell division phases also: prophase, metaphase, anaphase, telophase. Maximum you can see metaphase and anaphase phases, in fact. That is one thing. And in the light microscope, you can see the human cells, the cilia that is present. All of these should be stained with a color, then you'll be able to see properly different types of bacteria, different types of lysosomes, and their respective diameters have also been given. Sorry, is someone saying something? Okay, fine. There is no, Mr. Rishi, there is no notes as such for this. Whatever slides I'm like sharing, I'll just like, kind of share it with you. Is it okay with you? One of you just type in a minute.
Yeah, okay,
Yes,
ma'am.
ma'am. Yes, ma'am, it's okay with us. But, madam,
Oh,
could you please tell us where will you be sharing these all things?
I think you guys have a group with, MSc Microbiology, like I mean, MSc Zoology group, first years.
Yes, ma'am, there Kishore sir is the admin.
Yes. So add me
No,
into that
no,
group,
ma'am.
or one of you
Ma'am,
just like,
yeah,
yes. Can you type in a
ma'am,
message if
we
you
are not added to the group.
okay, whoever
Okay, okay.
request Kishore sir.
Yes,
Okay.
ma'am, we'll do that.
Right.
Like,
Right.
thank you.
So that case scenario, what I'll do is, once the class is over, I will just share my slide, whatever is there with me, with you. That's about it. Because I'm not, all this also has been taken from the internet. I'm being very honest and clear about it. Okay. So, and I do not have your textbook as such. So I will be teaching in general. So if there's a possibility, add me to the group. One of you put in a message to me, and I will just share all these, like, whatever slide I have, and whatever PDF or PowerPoint I'm teaching through. Is this okay with you?
Yes, ma'am.
Okay. How can you contact Kishore sir after the class gets over? I will just like share Kishore sir's number. Let me tell Kishore sir's number. I think some of you must be having Kishore sir's number,
Yes,
right?
madam, we do have. Can I text it in the chat group so that others
Yes,
get it?
please. Yes, please.
Yeah, ma'am, I'll do that.
Yeah, sure. Okay, let me continue with my lesson. So what can be seen through the naked eye? As I told you, the human egg, and then with the light microscope, with the having of resolution of 200 nanometers, you can see the cilia, the mitochondria and the bacteria, and with a much more finer resolution, that is 0.5 nanometers. So you're going to see the nuclear pores that are present because nucleolus has a nuclear pore in the middle connecting between them basically. That one, the centrioles, these are the smallest of the points that are present.
Excuse me,
The ribosomes and the globular proteins, etc. And when it comes to the basic components of a cell, you have the cell surrounded by the cell membrane or the plasma membrane, and you have inside them different kinds of organelles that are present. So the plasma membrane is the boundary of any cell that is present. What is it made up of? Lipoprotein layer. We have all studied this, but I still have to keep telling you in a little more detailed manner. So let's just be with it. So when it comes to the cytoplasm, it's the liquid that is present, basically having that is the liquid that is present, or the cytoplasm is, how do I say? It has cytosol, which is the intracellular fluid, or the ICF as it is known as. You have all the cell organelles that are present, the mitochondria, the Golgi complex, ribosomes, lysosomes, and then you have the cytoskeleton, which is the cytoplasm, wherein what is happening is, with the endoplasmic reticulum, you're going to connect it to the outside of the cell also. And when it comes to the extracellular fluid, the cell is present in a liquid medium. So that is your extracellular fluid, which is present. Let me just get it up. Right. And you also have in between the tissues, the interstitial fluid that is present. So what are we going to learn with the cell surface as such? You're going to see the structure of plasma membrane. What are the lipoprotein layers that are present? What are the carbohydrates that are present over here? And how is cell signaling taking place between cell and cell? And how are hormones actually important to us? So that is where you have your secondary messenger system and the cell signaling coming into place. There is it is a wholly there's one particular chapter for you that has been designated only for that, and you have the signaling pathways in it. We will come to that maybe once, in the future. What is the component of the cell membrane and the cell surface? Then what are microvilli, cilia, and flagella? I request one of you to actually just, open. Do you all have your textbooks? Can you just put it in the chat box?
yes, ma'am, we have.
You have the text. Okay. This is my number here. I'll just 4807301. So this is my number. You can have it. I'm Pranati. Next class, one of you in the group itself, if at all you can add me to that group, that will be very nice. Or I'll ask Risho sir to add me to the group. One of you, just personally, please text me the contents of the cell. I mean, the particular topic. Tomorrow's topic is what? Methods in cell biology, right? So, overview of the cell shape, type. I just want you to give the contents. You understood, no? The first topic, you remove the page, you get the contents. Only that part I want.
Okay, madam, we'll text you the headers of the contents.
Yes, please. Excellent. Take a book and read. Go. Right. So when it comes to the plasma membrane, is the border of the cell. That's what is being given. It covers the cell, shows like a dark parallel line, has both has two faces basically. One face of the plasma membrane is going to face your cytoplasm. The other face of the plasma membrane is going to face the extracellular fluid. So with the plasma membrane, what is going to happen is it is going to form a boundary of the cell. And this plasma membrane has again pores, has again signaling molecules to that, has again many other proteins on it, which will help in maintaining the position of the cell inside that particular tissue. At the same time, there are signaling proteins on the surface of the plasma membrane, which will help in interaction with the other other cells that are present next to each other. At the same time, the function of that particular cell, like I told you, if it is a pancreatic cell, it is going to give out insulin, glucagon, and so what is going to happen is it has to produce insulin. So this insulin has to be expelled out so that it goes to the rest of the body. So that is the particular function of that particular insulin cell, which is covered with plasma membrane. So in this manner, every cell which has its own particular function, it has to be done. So this interaction with other cells is done by the proteins that are present on the plasma membrane. Also, every cell requires energy that is being passed through the formation of glucose that is available in the extracellular fluid. Once you eat your food, that finally, after all the processes that are done, you're going to get glucose. Glucose has to get inside the cell. Or if the cell needs any other component, it has to get inside that particular cell. It is done through the plasma membrane. If you want any kind of ions, hydrogen ions or calcium ions, or if it is a nerve cell, you should have for nerve impulses, you have had calcium ion, sodium, potassium being going out, coming in. So in this particular manner, all of this is going to be controlled by the plasma membrane as such. So you can see it is a lipoprotein layer. In this you have proteins embedded in a phospholipid bilayer. And plasma membrane is not rigid, it is flexible. So what are they present? You can see these are the peripheral extra. See, this is the extracellular fluid. You can see the glycoproteins. So what is glycoprotein? Just split the word. If you're not able to understand, always split the word into two. You're going to get the answer. Glycoprotein. So it has a carbohydrate moiety attached to it. And you have the protein over here. This is the protein part. And these are the carbohydrate chains. When it comes to glycolipids, again, this is the lipid bilayer as such, and this is the carbohydrate chain that is present. Peripheral proteins are on the sides that are facing the extracellular fluid. Integral proteins, this is the word that you use, are present on the inside of the they are present inside the lipid bilayer as such. Again, this is also a peripheral protein, but facing the intracellular fluid. And in the middle of the phospholipid bilayer, you have cholesterol molecules also. So this cholesterol molecule, whenever there is a change in the temperature, because we have fluid dynamics that are being seen in the phospholipid bilayer, it is a fluid layer like I told you. So because of which this expansion contraction based on the temperature is going to be done by the cholesterol. And this is the intracellular phase of the membrane as such. And this is the transmembrane protein. Again, different kinds of transmembrane proteins are there. Peripheral proteins, integral proteins that are present, and transmembrane proteins as such. And when you see 98% basically is lipid. It's a lipid bilayer, phospholipid bilayer. So in this phospholipid bilayer, you have different kinds of molecules that are arranged in the bilayer. When I say bilayer, like I told you, it's a two-layer, one facing ECF, one facing ICF. Hydrophilic moiety are present facing each other. Hydrophobic tails are directed towards the center. See, simple. Hydrophilic, this molecule head is facing the fluid. Hydrophobic chains are facing each other because they repel water. You need this particular balance, otherwise you will have either more amount of water coming in, or that fluid balance of outside and inside is not going to be there if at all there is no hydrophobicity present. And what are the different kinds of lipids that are present? We have cholesterol, like I told you. This will actually help in making the layer, like the phospholipid bilayer, stable. And then you have the glycolipids, phospholipids, sphingolipids, and then you have different kinds. Again, biochemistry will come to that. And then you have the carbohydrate moiety that is present on the lipids again, because you need that carbohydrate chains also. Your hemoglobin and the red blood cell, the blood cell types is basically because of the different kinds of carbohydrate chains that are present. That is going to give you the different kinds of AB groups, in fact. When it comes to the membrane proteins, it is around 2% of the molecules that are present. Percentage is the amount of the molecules that are present, but they weigh a lot. That is around 50% of the membrane is the membrane proteins. What are they? Integral proteins. So in this again, you have the transmembrane proteins, like I told you, like you can see over here, okay, just basic picture. Cytoskeleton is here. This is another protein layer. You have a carbohydrate moiety and coming through that. So you have again proteins, different like you have the I'll tell you the serine, threonine, like these are the amino acids that are going to make the protein chains over here. In this, you will get to see that the different kinds of proteins are also going to help in your signal transduction pathway. Peripheral proteins attach to any one phase of the protein, and they are not going to be piercing through your transmembrane, through the membrane as such. And you have GPI protein anchoring proteins that are present basically, and then you have your I'm not able to remember the names as of now, but then you have different types of proteins. I'll just like kind of tell you the next class if possible. And where are they going to help you? What is the function of all these proteins? Why are so many proteins that are present? Like I told you, secondary messengers, that is cell signaling pathway, they're going to actually help you in. So you have this is a receptor. What is a receptor going to do? It will help. There is a chemical messenger which comes in, binds to this chemical messenger. Then this is going to send in message to the next part. And the protein itself is an enzyme. So once this protein is an enzyme, what will happen is once another chemical comes, this is going to break down. It can be a channel. Hydrogen, sodium, potassium ions, all of these can pass through. Entry or exit, it depends again. You have both types, you have gated channels as well as normal channels that are present, non-gated channels. So that is with a gate means, it will need energy for the channel ions, for the gate to open as such. You can see them in your stomach, and the what do you call it, the nerve transmission. And if you see the channel protein constantly open, allows solutes, water molecules also to pass through. Gated channels, you can see, allows the solutes only through certain times. You do not need insulin being produced or being pumped into the cell all the time. Insulin will come only when there is glucose available. Simple. Cell identity markers. What are they? They are again glycoproteins. Carbohydrate moiety is present. It's a protein which is present. So that what will happen is it will identify your immune system. It will identify the foreign particles that are present inside the cells. For example, if there is any kind of foreign, the bacteria that is present. So it is non-self. So that identification should be able to be done by the cell. So this particular glycoprotein helps in it. CAM is one more. This is cell adhesion. So CAM is basically cell adhesion molecules. They bind each cell. So when it comes to membrane cells, like I told you, receptor, secondary messenger. Sorry. I'm so sorry. Just give me a minute. Right. So again, channel proteins, ligand gated, voltage gated, mechanically gated. That is, ligand gated means whenever there is any kind of hormone or another chemical messenger that is coming over. Voltage gated when there are any ions that are present. Sodium, potassium, or a bicarbonate ion, which is HCO3 minus, sodium plus hydrogen plus. So all of this will create a charge. So it's a voltage gated channel. Mechanically gated channel, because of any kind of stress on the cell, it is going to be open. So if there are more number of any kind of ions or any kind of molecules that are present inside the cell, this mechanically gated channel is going to be open. When it comes to the membrane proteins, again we have different types, that is carriers, cell identity demarkers, and the cell adhesion molecules. You have seen that. Right. When it comes to the secondary messengers, you have the G protein coupled receptors that are present, and they activate something called as a GTP. So, which is your guanosine triphosphate. This will release a whole lot of secondary messenger or cell signaling that takes place inside. Finally, it will lead to three outcomes. One is changes in the DNA, or it will cause cell signaling, or it will cause cell death. These are the three outcomes whenever there is a secondary messenger taking place. So we can see the G protein relays the signal. So what is going to happen is there is going to be a receptor on the cell, and this receptor activates the G protein, which is present on the periphery of the cell. It's near your ICF, intracellular fluid, on the other side facing in the plasma membrane. This relays the signal to adenylate cyclase, conversion of ATP. So there is energy involved over here to CAMP, cyclic adenosine monophosphate, which is a secondary messenger. This will activate other kinases. And whenever there is a kinase action, if you have read enzymology, you'll know that kinases are phosphatases. So what is going to happen is addition of a phosphate group is taking place whenever there is a kinase enzyme involved. So addition of phosphate groups to the other enzymes, because of which these enzymes will work on different kinds of action depending upon what the first messenger, that is the epinephrine, has done, and what is the overall necessity for that particular cell. So your drug action, which will it will either modification of the at the cellular level, that is your DNA is going to take place, or it will cause cell death, or it will cause what do you call it cell division to take place. This is the final outcome of a secondary messenger. So you can see over here secondary messenger system. First messenger, then adenylate cyclase is over here, then conversion ATP to CAMP. CAMP inactive kinases are present and they are going to be activated. This inactive enzyme which is present is again because of the activated kinases, they activate the inactive enzymes that are already present. So then you have a different kind of metabolic effect. When it comes to the glycocalyx, it is a fuzzy coat which is external to the plasma membrane basically. I would like to actually see if at all I've got another. So not this one. I have 3 or 4 slides basically. So I'm you can just have a look at it. So structure functional unit of living organisms discovered in 65. Cell theory is all the living cells arise from other cells. You've seen that. Prokaryotic cells, eukaryotic cells, basic distinction or so prokaryotic cells do not have a membrane bound, they do not have a nucleus as such, and their membrane bound structures. This is the bacteria. Eukaryotic cells, nucleus, and every membrane, mitochondria has a membrane as such. Endoplasmic reticulum has a membrane as such. So this is what they mean by membrane bound organelles. And they're all higher organisms. Prokaryotic cells, like I told you, circular, no histones present. This is the basic cell structure as you can see. Cell wall is present, has ribosomes. Again, ribosomes are of different types, like I told you, no? So, when it comes to the eukaryotic cells, you have membrane-bound nucleus, DNA, histones, all of these are present. And when it comes to the structure of the cell, you can see that this is the cytoskeleton. I think this is a good picture that I'm sharing with you. This is the proteasome. These are the microfilaments. Let us just start with the outer This is the cell membrane or the plasma membrane. This is the nucleus that is present. Connecting to them is the endoplasmic reticulum. If there are proteins lined on the endoplasmic reticulum, it is known as rough endoplasmic reticulum. That is your if there are any ribosomes attached to the endoplasmic reticulum. Okay. So this rough endoplasmic reticulum is involved in cell division and is involved in production of proteins, protein synthesis. If the endoplasmic reticulum is not attached with any kind of ribosomes, 70S, 80S ribosomes as such, then it is known as a smooth endoplasmic reticulum that is involved in lipid synthesis and lipid packaging, and we'll come to that later. Now, when it comes to the nucleus, we are again talking about the eukaryotic cell over here. You can see the chromatin, the different kinds of nuclear pore complexes that are present. This is the nuclear envelope. And within that, you have the nucleolus. So, you have the endoplasmic reticulum and the nucleolus, and then you have the nuclear pore complex, again, having open, which opens and closes because of the presence of different kinds of proteins in it. Again, you have glycogen granules which are present inside the nucleus, and this is the cytoplasm, which is a fluid. And you talk about the cytoplasm in whole except with the nucleus. The liquid that is present in the nucleus is known as nucleoplasm. Just that word difference is there, please try to understand. And then there are ribosomes. You have 70S, 80S. We have seen that in earlier, in the sense, in the your undergrad also. The Golgi complex, packaging of lipids takes place. And then you have different kinds of microfilaments present. Then when it comes to the skeleton, you have the microtubules, you have the microfilaments and the intermediate filaments, each with their respective function. You have microvilli. Microvilli especially present in cells which have to be passed on. So, just a second, somebody. Right. And when it comes, like we have already seen the plasma membrane just a little while ago. And then you have different kinds of secretory vesicles which are released from inside, which comes and finally goes, attaches itself to the plasma membrane and releases whatever it has to release. Lysosomes, cell destruction molecules, as they are known as. Then the smooth endoplasmic reticulum. You have the peroxisomes, glyoxysomes, the mitochondria, which is the powerhouse of the cell, and the microtubules that are present. So, difference between prokaryotic and eukaryotic cell, you can just see basic ones, size differences there. Prokaryotic cell unicellular, eukaryotic cell multicellular, single membrane, bilayer membrane is present, and no nucleus, linear DNA. All of these are the basic differences you can see. Prokaryotic cell has the 70S type of ribosome, wherein the components are of 50S and the 30S type. Ribosomes are like on the eukaryotic cells are 80S, which is like 60S and the 40S types. Can anyone tell me how you're going to actually find out the sizes or the weight of the ribosomes? Maybe in the next class, one of you can message me. Cell division is going to be by fission. And over here in the eukaryotic cell, it is mitosis. Of course, meiosis also is present. So yes. And when it comes to the cytoskeleton, it is not present. You have flagella that are present. Prokaryotic cells, bacteria. So you have flagella present. Rickettsia is one of the organisms which are prokaryotes. Eukaryotes, you have fungi, plants, animals, whatever, the rest of them all uh, that is present. I think, there is no point in me asking, for you to tell in the next class. It is going to be decided, taught here itself right now. So you see, subcellular organelles, if at all I want to isolate ribosomes, if at all I want to isolate the nuclei, or if at all I want to isolate any parts within the cells, how am I going to do that? First, what I'm going to do is, the cells that are present, I'm going to take the tissue and then disrupt it or put it in a homogenizer test tube. And I'm going to, so I cannot just take a small tissue piece. If at all I want, I'm going to take out the liver piece of rat. So if I'm going to do that and I want only liver cells and the enzymes that are present in it, or the and or the different kinds of whatever I want in from there, the ribosomes of the liver cells, for example, or the enzymes that are present inside the liver cells, or the catalase enzyme if at all I want, or or any kind of alpha fetoprotein enzyme if I want. What am I going to do? I just can't take the tissue, liver tissue, put it in the homogenizer and then start using a motor pestle. It's not going to work like that. So I need a solution to suspend the cells first. It is going to be an isotonic solution, meaning it should be in the particular solution should be present in something which is it is compatible to, which has already been present. So it's going to be sucrose, which is around 0.25 molar. And then you're going to have it in a buffer solution. So you have different kinds of buffer solutions available also. You have TEA buffer, and then you have different kinds of buffer solutions. You're going to maintain the pH that is present. You can use bicarbonate buffer. And then once you do your practicals, you will get to know different kinds of the buffer solutions are also that are present. And then I'm going to see that if at all I'm going to isolate the organelles, then the sedimentation coefficient that is present should be the same or similar. That is how dense they are. Based on the density, I'm going to isolate them. And then I'm going to put in markers, especially, so they are going to be your enzymes, proteins again. So how pure is the subcellular organelle that I have isolated? So if at all I want to isolate different kinds, that is, my nuclei or mitochondria, Golgi complex, endoplasmic reticulum, I'm going to take the homogenate and the plasma membrane, I need to disrupt it. So what am I going to use? Sodium potassium ATPase. So 10 minutes, 960 G, I'm going to use an ultracentrifuge for it. So I'm going to get a pellet, which is going to be having the nuclei, the DNA polymerase that is present. This is the supernatant. I'm going to collect it again at 25,000 G. For 10 minutes, if I do, I'm going to get another isolated pellet over here. This pellet has mitochondria, lysosomes, peroxisomes, ATP, phosphatase, cathepsin, all of this. So this is the homogenate. These are the organelles that are going to present inside the pellet and the supernatant. And then these are the enzymes, marker enzymes that are going to use to find out whether it is the same. And again, if I centrifuge it again at 34,000 G for 30 minutes, I'm going to get the isolate of Golgi complex. For this, I'm going to use the transferase enzyme. And again at 1,05,000 or what do you call it, G for nearly 3 hours plus, then I'm going to get the supernatant, which is only the cytosol. The pellet again has, and I'm going to use different kinds of enzymes, which is your lactate dehydrogenase, glucose 6-phosphate dehydrogenase, all these enzymes to find out presence. So when it comes to the plasma membrane, like I told you, has nucleus, cytoplasm, subcellular organisms. Nucleus control center. It's very it's there. It's usually in the center of the cell. Single nucleus is present, except in some cases you have your RBCs. In your RBCs itself, you have different kinds of cells, right? Eosinophils, neutrophils, basophils. Eosinophils have different kinds of nucleus, more nuclei. And then the nuclear envelope is of two membranes, outer and inner. The inner nuclear envelope faces the nucleolus or the nucleoplasm. Outer nuclear envelope faces the outer membrane faces the cytoplasm. As it's similar to that of your plasma membrane. The continuation or the invagination of the plasma membrane inside forming the endoplasmic reticulum up to your nucleus, your nuclear membrane, and then inside that you have the nucleolus, again connected through different kinds of nuclear pore complexes that are present. So you can see outer in continuity with the endoplasmic reticulum. Inner perinuclear membrane is also present. This has the nuclear pores and the nuclear pore complex to present in it. And it has a huge central opening. You have different kinds of protein arrangements in the circle in the form of four and four. Four proteins on the top, four proteins in the bottom with a channel in the middle. And then control of the proteins RNA is going to take place in this manner, production of DNA, and then when your transcription translation takes place, you have the nucleus playing a very huge role. When it comes to the nucleus, what does it do? It has the DNA, that is your chromatid. What is it used for? Protein synthesis, ribosome synthesis. So ribosomes in turn produce your protein synthesis. That is where you have your transcription. Then you have the protein synthesis translation again playing an important role. Nucleoplasm, different kinds of enzymes are produced. And you have your example is your DNA polymerase. And it is the site for DNA replication. Replication of DNA means production of a duplicate or a similar DNA. Depends. You cannot always get the duplicate DNA. So if there is meiosis involved, you're going to have crossing over chiasma and all of that taking place, because of which you're going to get your hybrid DNA. Otherwise, DNA in case of mitosis, it is going to be duplicated. So DNA replication. Then RNA is going to be synthesized. This RNA goes out and then you have the translation for proteins. So you can see over here, nucleus has the DNA. This is the nuclear pore complex like I told you. And this is going to be where all the cellular activities are going to take place, and then production of ribosomes from the nucleoli is going to take place. Mitochondria, we are talking about different kinds of cell organelles present. So if you see mitochondria elongated, it can be rod-shaped also. Again, two membranes are present. It can be 100 or 1000. It depends upon the activity of that particular cell. If you need mitochondria to produce more cells, then you're going to have more number of mitochondria present. Like I told you, again, mitochondria, the outer layer is going to be continuous, and it is lipid. It has lipids or more number of lipids present, allows all the smaller molecules to present. And then you have an intermembrane space over here, which is present. And then you have an intermembrane space here as well. Inner is proteinaceous in nature. So again, you have one particular component called as the cardiolipin, which is present. It is not permeable and it folds in the form of crystal. The particular necessity for it to form in this way is that, so that it can accommodate, so that the surface area becomes less. So, I'm sorry, it increases the surface area for the production of ATP. Because currency of the cell is finally ATP. So what is mitochondria going to produce in the end? The complete end product is ATP. Whatever glucose is being consumed by the cell goes inside and ends hit the mitochondria. Whatever biochemical process is taking place finally, glycolysis, then you have your Krebs cycle taking place. Their end products are ATPs, if you noticed. This ATP is going to go into the electron transport chain. Where is the electron transport chain taking present? In the mitochondria. So this is where your ATP synthesis is taking place. This ATP is going to be stored, is going to be utilized by every other component of the cell. So this ATP goes, because for the smallest of the biochemical reaction, you need ATP. So ATP is the cell currency. Sorry. Finally, the intermembrane space has your enzyme, which is adenylate kinase. Now, the mitochondrial matrix has a circular DNA present. It has ribosomes and it has different kinds of enzymes. And your mitochondrial DNA, whatever is present, is going to be Completely. There is no doubt about it. Maternally inherited. So because of which, whatever mitochondrial disease you're talking about, for example, lupus, I think, lupus is a, maternally inherited disease, if I'm not wrong. And other mitochondrial diseases that are present, I'll just like tell you the name. For example, you have different kinds of mitochondrial diseases that are actually inherited from the maternal side itself. It cannot be paternal as such. So it is again going to affect all your wherever there is mitochondria. So obviously all of the what do you call it, you have wherever mitochondria is present, so no organ is exempted. Example is your mitochondrial encephalopathy, then you have your Lay disease, you have a different kind of epilepsy that is present, then you have Leber hereditary, of which is an optic neuropathy, so there's a problem in the eyes. All of these are mitochondrial diseases, and they are maternally inherited through the mother only. So what exactly is the function of mitochondria? Like I told you, energy production, all your metabolic pathways, your Krebs cycle, urea cycle, fatty acid oxidation, whatever is taking place, beta oxidation, all of it takes place in your mitochondria. Now, when it comes to chloroplasts, chloroplasts are those cells that are present only in plants. Helps in the conversion of energy, which is coming from the light, into ATP. This is done by photosynthesis. So, like I said, basic. If you see the magnified part of this particular cell, mitochondrial membrane, you're going to see all of them are enzymes that are present. These are the ribosomes that are present. And you can see the matrix, the cristae that are present. Outer, inner, intermembrane space. Now, when it comes to ribosomes, you see they are the sites of protein synthesis. They are of two units basically, a large subunit and a smaller subunit. In the case of prokaryotes, you have 70S, which is again made up of two components, a 50S and a 30S. Eukaryotes has two components again, 80S. So it is known as 80S subunit. It has a 60S and a 40S subunit. And when it comes to the endoplasmic reticulum, it is a network which is going to connect. These are the roads, or it is a network that is going to connect different kinds. It connects the plasma membrane to the nucleus. It is going to connect Golgi complex to the rough. So again, like I told you, two types, rough and smooth. The rough has the ribosomes which are present, because of which it is known as rough endoplasmic reticulum. Smooth because it has it does not have ribosomes attached to it. Rough endoplasmic reticulum function, protein synthesis. Smooth in the production of different kinds of phospholipids, which are going to go and get attached to the cell membrane or the plasma membrane. You have the production of cholesterol and production of the different kinds of hormones. All of it is taking place in the smooth endoplasmic reticulum. And it is the site of glycogen metabolism also. Glucose is going to get converted into glycogen. So then what is going to happen is, whenever that is taking place, removal of a phosphate group is going to take place, because of which release of free glucose inside the blood. So glycogen metabolism is wherein the glycogen is going to get converted into glucose. So whenever there is excess of glucose inside the body, first thing is what is going to happen is you're going to get it converted into glycogen. It can be in the muscle tissue, it can be in in other sites as well. So and whenever the body requires the particular glucose, what is going to happen is this glycogen is going to get converted into glucose again and get released, and goes to wherever it has to, wherever there is a necessity of glucose. This glucose is again finally in the mitochondria getting converted into ATP. So you have sarcoplasmic reticulum. It is a term which is present. Sarcoplasm, myoplasm. Basically, these are present inside the the endoplasmic reticulum, which is present inside the, the muscle cells, is known as sarcoplasmic reticulum. So what is it going to do? Take out, or it's going to store, it's got a specific function to it, stores the calcium ions. So this calcium ions, whenever they are stored, what happens is, whenever there is a necessity for the muscles to contract, relax, so muscular contraction is going to take place with this. So you have different kinds of cells, right? Myotonin, and then so all this muscular contraction is because of the release of the calcium ions. And if it is present in the liver, in the kidney, then that is where, drug detoxification. So liver is the place wherein all the cells, whatever toxins get into the body, and then you have to, purification is going to take place. Kidney, it purifies the blood. Then whatever, all of it go, and your complete detoxification is going to be done in your liver, kidney. Spleen, graveyard of RBCs, wherein after 120 days of the RBC life, it is going to go into the spleen, and then there, that is where, your cell, the lysis of RBCs takes place. Now, what is the function of Golgi complex? Consists of different types. Again, this is around 3 to 20 cisternae that is present, and then the sorting, it is the packaging of the proteins. So production of proteins is one thing, but sorting, packaging, where should it go? Where should, is there any kind of, the glycogen layer that has to be added to the protein to form a glycoprotein? Or should it get a lipid layer? So whenever there is a production of lipoprotein, lipid being added to, it is all going, all this kind of modification is going to take place in your Golgi complex. What are lysosomes now? This is on the another cell organelle that is present. It is again enclosed, and this is it comes out from the Golgi complex. It is a tiny organism. Like I told you, they are the suicidal organelles that are present inside the body. What do they consist of? They have enzymes in them. Enzymes again are proteins. So what do they do? Digestive enzymes. Then you have cell lysis whenever there is apoptosis to be taking place, or you have different kinds of cells. All of these are present in the lysosomes. So what does it do? It kills the cell from within. So, and also phagocytosis. If there's any bacterial cell that is going to come smaller than your normal cell, and if that has to be engulfed, it is going to be done by lysosome. And your apoptosis or autophagy with the help of lysosomes. Again, you have different kinds of diseases. If there is any abnormality in the lysosomes that are present, or in the storage of the lysosomal enzymes, or there is any deficiency or absence, you have different kinds of diseases such as Niemann-Pick and Gaucher's disease. Peroxisomes, catalase function, oxidation of all the amino acids and different kinds of lipids that are present, long chain fatty acids that are present, and whenever there is hydrogen peroxide present, all of it is going to be taken care of by peroxisomes or micro bodies. And if there is any deficiency or a problem in the peroxisomes, then it is the Zellweger syndrome. Glyoxysomes in the plants are also present. They have different kinds of lipids in them. Cytoplasm, we have already seen over here. It is basically the site for many metabolic pathways as such. So it again has different, the cytoskeleton as such has protein filaments. It's anchored to the cell membrane or the plasma membrane. There are three types of proteins that are present on it. The microtubules that are present, intermediate filaments, and the microfilaments. It is the structural framework that is going to be there, takes or helps in the uptake of materials inside the cell, and the movement of different cell organelles, then muscle contraction, cell signaling pathway, all of it is going to be because of the cytoskeleton as such. Cell division also, so the cytoskeleton is going to play an important role. So you have the microfilaments that are present. You have actin, tubulin, myonin that are present. and then you have the intermediate filaments. So based on the diameter, you have the different names as such, and based on the filaments that are present, basically. So it has actin over here, and you have the tubulin. That is why it is a microtubule, and it is hollow, and the diameter is around 25 nanometers. So microtubules are big. So they are hollow. They have actin and tubulin proteins that are present, which are again of different types like alpha and beta. And it is the largest of the cytoskeletal components that are present. Where are they present? These microtubules are mostly present in the axon of the nerve cell, the dendritic cells in the nerve cells. Microtubules, structural support and to maintain the particular shape of the cell. And through the microtubules, there is a hollow part, right? You have movement of different kinds of organelles that are present, different kinds of vesicles, and also if at all there is any kind of cell or the hormones are to be released, etc. It is exocytosis that is going to take place through the help of microtubules. It is also going to be helpful in the formation of a mitotic spindle. That is during your, the microtubules are going to help in your mitosis, in the formation of the spindle fibers. And movement of cilia and flagella is also because of the tubules. So you have actin, you have dynein, different kind, and then tubulin, different kinds of proteins are going to be making your cytoskeletal protein as such. In case there's a disorder in this, it is going to be primary ciliary dyskinesia, and then you have upper and lower respiratory tract infections and male infertility because of all of this. You have intermediate filaments, like I told you, actin, you have intermediate filaments, microtubules. So in them, again, you have different kinds of, again, made up of proteins. Our whole body is made up of maximum proteins. Then you have carbohydrates, which are going to be stored for storage of energy, and lipids, which are going to give you your the cushioning effect as such, your adipose tissue. So you need proteins. Hence, if you notice, that is the primary reason your diet should consist more of proteins, less of carbohydrates and lipids, because everywhere you need proteins to sustain. You need proteins for energy. And so let me be back to the lesson. Intermediate filaments, again, polymers of long rod-like proteins present, again made up of different kinds of proteins, keratin, neurofilaments, etc. What do they do? Mechanical support. So different places you have different kinds of filaments making up or providing that particular mechanical support. So help in intracellular attachment. And this is present usually, the intermediate filaments are present in your neurons. So, and they're also present in the skin and the hair. Microfilaments, they're the smallest, the thinnest of the cytoskeleton or the structural framework that is present. What are they made up of? Actin filaments. So actin has again different types. So we have alpha, beta, gamma type, actin in fact. So when it comes to that, these microfilaments are made up of beta and gamma. They form the mesh that is present right beneath your plasma membrane. They help in absorbing the stress. So that is why they're known as your stress fibers. What do they do? Basic mechanical support for the strength and shape of the cell. So if there is any kind of let's say um, uh, stress coming and falling on the cell, that is when you need these microfilaments, which are going to give you that particular say something like your bouncy ball. You have the uh, kids play, right? The trampoline has different kinds. So if you just imagine this microfilaments forming that particular trampoline layer beneath, having that the square kind of network this is present, which is present underneath, then you can imagine. So they provide the basic shape and strength to the cells. Examples of the microfilaments are the microvilli. So, and they provide the shape involved in muscle contraction, cell division, cell locomotion. So, wherever there is necessary, and the microvilli inside the gut actually help in pushing the food particles down, or like, what do you call it, help in pushing of the molecules into or outside, depending upon like, the necessity. So finally, when you look at it at the larger picture, you get to see that mitochondria is for production of ATP, different kind of metabolic pathways. Your Krebs cycle, glycolysis within the cytoplasm. Krebs cycle takes place in the mitochondria. Fatty acid oxidation, that is your beta oxidation, takes place in the mitochondria. Urea cycle takes place in the mitochondria. Nucleus has your DNA in it in the form of chromatid. Ribosomes, assembling of the proteins, and then you have the endoplasmic reticulum, wherein you have the protein translation taking place. So protein synthesis is one step, transcription, then translation, then folding. Protein folding is very important. If there is any kind of problem in the protein folding or the misfolding, you're going to have diseases. Example is your protein folding, misfolding diseases. Alzheimer's is one, and your Parkinson's disease is one. If there is any kind of protein folding, misfolding, because of that, it causes the diseases. So it is very important. You have your heat shock proteins that are involved in it, and you have chaperones that are involved in it. So different kinds of molecules. Anyway, transport of proteins to different places. Once the protein is produced, for example, your protein is produced, it has to go to your plasma membrane. It cannot just stay inside the cytosol as such. So protein transport also, endoplasmic reticulum is playing an important role. Golgi complex, all the cell proteins, whatever is there, it has to be delivered. It is going to be done by your Golgi complex. Cell packaging is going to take place. Transport network is your endoplasmic reticulum Golgi complex. In the case of plants, you have chloroplast, wherein photosynthesis is going to take place in it. Again, energy production. Vacuoles for storage, secretory, all the enzymes that are present. Lysosomes, digestion, cell death or apoptosis is going to be because of lysosomes. Peroxisomes for your basic toxic substances to be broken down for your oxidation. And your antioxidants, basically they are the ones which are going to be helpful. Glyoxysomes, glyoxysomes, so what is going to happen? Fatty acids are going to be converted into carbohydrates. So plasma membrane, again, when it comes to the most important structure, or it's the lipid bilayer. That is what Davson and Daniel, scientists, had actually said. And it has a fluid mosaic model. That is what Singer and Nicholson had actually described in their paper. And that is because it is fluid and mosaic. Mosaic in the sense because you have proteins which are floating, or proteins that are present with cholesterol, which is there, again it is a lipid present in the bilayer, lipid bilayer, phospholipid bilayer. So the lipid bilayer has phosphate and proteins attached. You have carbohydrate moieties, and all of these are floating. They are present, they are floating. It is fluid because when there is heat, like I told you, the cholesterol molecules help in expanding. So it is not exactly a rigid structure. So what does it what is it made up of? Lipids, proteins, and carbohydrates. Structure, like I told you. Integral protein, that is your transmembrane protein. And you have the glycoproteins having a carbohydrate moiety. Glycolipids. So carbohydrate moiety present on the lipids. And then you have the cholesterol molecule. So this is what? And this is a hydrophobic moiety. This is the hydrophilic moiety. What are the membrane lipids? 75% phospholipids, 20% cholesterol, 5% glycolipids. Again, amphipathic as such, polarity. So polar and non-polar. And what do? Act as a permeability barriers. They do not allow everything and anything to come inside. So you have channel proteins, that is through which only the components can come inside. Ions, molecules, glucose molecules, if it has to come inside. So you need to have an insulin receptor over here. So because of which that insulin receptor, if this is an insulin receptor, the glucose molecule will come here. Then the insulin receptor has to undergo change, because of which the glucose molecule will get inside the cytosol. If it is an ion channel, then the ion, sodium, potassium, hydrogen, whatever ion is necessary, it goes inside. So ion channels can be gated, non-gated, mechanical, that has also been seen. And this is the extracellular. This is also a transmembrane protein, which is like present. You have seven protein layers, etc. Like, different kinds are there. We'll come back to it again. So this is necessary for fluidity of the cells. Phospholipids, head is made up of a polar phosphate group that is present, non-polar hydrophobic long chain fatty acids. So fatty acids are short chained, medium chained, and long chained fatty acids that are present. They are the lipid part. So you have glycerophospholipids, you have sphingophospholipid, you have phosphatidylcholine, inositol, then plasmalogen, sphingomyelins. All of these are the lipids that are present. If you have read the chapter called lipids at any point of time earlier, then you will know all of these are the lipids that are present. So they are also having a different kind of names like C9. So like, and so what is going to happen is all of these are arranged in the phospholipid bilayer. This is the protein that is present over here. This is the fatty acid, and lipids are usually, or they are hydrophobic in nature. So this will balance the membrane, and they are not going to be linked or attached to the neighboring phospholipids by different kinds of bonds. So what, because of which there is only a lateral movement as such. And cholesterol is going to be present in between, and it is amphipathic, basically, again weak, and it has other lipids attached. This will contribute to the stability of the plasma membrane, but at the same time it can alter the fluidity of the plasma membrane. Then fatty acids, usually cis fatty acids are present. So same kind of fatty acids, which are like arranged together. So that is why it is a cis fatty acid. So they increase the fluidity. Glycolipids present on the outside of the cell membrane. And what are the different kinds of membrane proteins? Integral, peripheral proteins, and the transmembrane proteins. Integral proteins, either they are totally inside the protein, inside the completely embedded inside the plasma membrane, or peripheral, that is they can be outside the plasma membrane. If they are either over here outside facing the extracellular fluid, or inside facing the intracellular fluid, they still are known as your peripheral proteins. Integral proteins are completely embedded inside the plasma membrane. I hope you have understood this basic difference. And then they usually glycoprotein, that is having a carbohydrate moiety attached to it. Peripheral protein, like I told you. So this is the basic structure you can see. Integral protein, these are the cholesterol molecules that are present. This is the glycoprotein, you can see it's a peripheral one, like, and you have the carbohydrate moiety attached to it. This is the ECF, this is the ICF. So what are the functions? Transmembrane proteins are ion channels or transporters. Peripheral proteins will be receptors. So they help in your signal transduction pathway. Enzymes, they undergo changes. So these are the functions of the proteins that are present. What are the membrane carbohydrates that are going to be present? Usually they are going to be attached to either the lipid or the protein. So they are going to be glycolipids or glycoproteins. So what are they? The carbohydrates that are usually attached are glucose molecules or galactose, mannose. So what are they going to be attached to? So if at all they are attached with an acetyl group, it is known as N-acetyl glycosamine. So you have the N and the piece, N's, right? So based on where they are attached to the protein, they are having a name. So N, amino group, if it is attached to the amino group, it is the N-acetyl glucosamine. So it has an acetyl group to it, it has a glucose group to it, and it is attached to the protein. So that is why it is known as N-acetyl glucosamine. I hope this part is clear. When you see a particular name, don't get confused as to why there is a particular, N attached to it or O attached to it, or if there is a you have different superscript to it. All of it. You just like split the word, you'll get the answer. Proteoglycans that are present on the outer surface. Glycocalyx layer has the carbohydrate layer that is present over, usually present in the uh, gram positive bacteria, if I'm not wrong. So, what are the functions? Imparts the charge to the bacteria, repels other particles. So, there's a complete glycocalyx present, helps in intercellular attachment. So, and it acts as a receptor. Then you have different kinds of markers. They are your, they give the basic identity. And your antibody processing is also because of the carbohydrates that are present. All your antibodies have different kinds of carbohydrates because of which they are named your um, so the antibody processing is because of this. Then specific features or special features, you can see it is a fluid, it is a sea of lipid bilayer. It is asymmetric in nature, fluid in nature, anchored to the cytoskeleton. So I'm not going to go into this particular slide again. Functions, semi-permeable barrier. We have gone through this again. Receptors for hormones, recognition site for antibodies. So you can see when it comes to the cell membrane, what is happening over here. Permeability. So all the ions are coming inside, acts as a boundary. And then controlled metabolite transport. So glucose metabolite, if at all any molecule has to come inside, if at all any secondary messenger or a hormone has to come inside the cell, it is going to go through this. So, and then signal reception transmission. So your CAMP pathway, there is one CAMP pathway, that is your cell signaling pathway takes place. So any kind of receptor is present, it comes inside. This leads to your cell signaling pathway. Enzymatic reactions also, this acts as an enzyme. So if A plus B, if the molecules are present, this enzyme, if the present on the cell membrane, is going to help in the conversion and production of CMB. Cell adhesion, and contact with other cells, and at the same time structural framework or anchor for the cytoskeleton. Transport across the different cell membranes also is seen over here. Now, you have different kinds of transport. Is your passive transport, active, and bulk transport, which is your macromolecules. So simple, diffusion, facilitated diffusion. Active is wherein your energy is being involved. Bulk transport is exo and endocytosis, wherever is necessary. Again, over here, you have seen uniport, co-transport. So you have your, transportation of the glucose in your red blood cells. So you have a GLUT. GLUT is the protein that is present. So GLUT transport as it is known as. Co-transport, you have sodium and glucose coming together. So co-transport. Antiport, wherein what happens is if sodium molecules come inside, the hydrogen molecules go out. So sodium hydrogen or sodium potassium, they both they are the antiport. Or example is your sodium chloride and bicarbonate exchanger. So whenever chloride molecules or chloride ions come inside, bicarbonate ions get out. So different kinds are there. Again, passive transport is simple diffusion. So you have different kinds. Yeah, you have your gases, water molecules, neutral polar molecules, lipid soluble molecules, all of it coming in. Channels allows only one ion. So example is potassium channel. So more number of potassium channels are there as compared to your sodium ions. Osmosis, diffusion of water that happens. So movement of water is going to take place against from area of the lower concentration to area of higher solute concentration is going to take place. That is your basic osmosis. And where is it going to be used? Osmosis has to take place. Otherwise, it is going to have an effect. What is it? Whenever there is no intake of water, dehydration. So you see it in the form of a decreased output of urine. Osmosis is one of the most important factors which is taking place in every cell, especially more in the kidney cells. And second thing is, in case there is any kind of swelling everywhere, you see there is retention of water inside the body, decreased urine output. So in the kidney, having people having kidney issues, you see that their feet are swollen, but their urine output is less. That is because all the water is being retained. That is known as your edema, swelling. So water retention. This can be different because of different issues as such. But you basically see that is because of the osmosis not taking place properly. Edema because of another issue, that is hypoalbuminemia. So you have albumin, globulin, different proteins that are present. So if at all there is less amount of albumin, so it causes edema, tonicity, and effect on the red blood cells. That is also where osmosis is important. So isotonic solution, same solute concentration as a solvent concentration. Hypotonic is when what will happen is this concentration of water is more and less inside the cells. So the cells, because of osmosis, absorbs water and bursts. Hypertonic, what happens is all the water is going to come out of the cells because the solute concentration is more. Normal RBC undergoes hemolysis and crenation. Facilitated diffusion, you see different kinds. Simple diffusion, channel mediated, facilitated diffusion, carrier mediated. So you, with the help of different kinds of carriers, this carrier has the ion attached to it, and then this is going to be facilitating the ion to get inside or outside the cell, because of which it's known as carrier mediated diffusion. Transport again is of two types, primary and secondary active transport. So you can see hydrolysis of ATP or sodium potassium pump. All of this come under the transport systems. So three sodiums move out of the cell, two potassium cells move inside, and in all of this, because it is an active transport, there is involvement of ATP molecule. Energy is required. Where is this active transport used? So to maintain the intracellular sodium potassium concentration. So this maintains in the electrochemical gradient. You have the nerve and the muscle cells. So nerve transmission is because of this sodium potassium pump. And inside your stomach, especially for transportation of sugars and amino acids exchange, calcium pump in the muscle contraction. Secondary active transport, symport, antiport, same symport, antiport is exchange of different ions taking place. So symport is also known as co-transport. So you have both sodium and glucose coming simultaneously inside. If at all there is any issue, you have diseases formed. So cystinuria, Hartnup disease is one of them. And you can see, this is a basic example. Sodium is being, and then along with the sodium, you have the amino acids, di and tripeptides. So this is a sodium dependent co-transport. This is an example of antiport. You have three sodium coming in, three sodium going out of the cell, two potassium molecules or two potassium ions coming inside the cell. So antiport system. This is the facilitated diffusion, wherein you have the amino acids, tri and dipeptides with the help of the carrier will come out. This is all taking place in the lumen of the intestine. This is the sodium glucose symport system. Antiport is sodium hydrogen exchanger, which is in the kidney cells, that is your nephron cells, renal tubules, and inside the cardiac muscle, you have the calcium and hydrogen being exchanged with the consumption of ATP. So example of symporters and antiporters at the same time. Again, you have different ion channels, transporter present, and at the same time you have the ATP powered pump. This is the ECF, this is the ICF, and then you have the basically it is the closed channels. Whenever presence of or absence of ions are present, it is open, close accordingly. Transporter wherein ion molecules. So this, all these, whatever you see within the membrane, they are all channels, pumps, transporters, they're all proteins. This is the phospholipid bilayer that is present. Unipolar, only one particular ion will come inside or molecule will come inside. Sympolar, you have a glucose and a sodium. Example. Antipolar, sodium, potassium, chloride, bicarbonate, all of it. One goes out, the other comes in. So basic differentiation. Bulk transport, wherein the membrane is going to be opened, and then you have many macromolecules coming inside. So again, it is ATP dependent, and it needs even calcium ions. So endocytosis is the wherein you get to see that it is wherein the cells are being eaten away, engulfing of the different molecules by the cell. So two types again, phagocytosis and pinocytosis. Phagocytosis is cell eating. So with the help of lysosomes, large molecules are ingested. Example is bacteria. So the white blood cells, they engulf the bacteria. So that is phagocytosis, cell eating. Pinocytosis is wherein the fluid inside the cell, that is the cytoplasmic fluid, is going to be taken into the cells. This is going to be taking place in all the cells. Example is your protein uptake. So this is how endocytosis is going to take place. You can see this is the cell or the plasma membrane. You have the lysosomes that are there. Bacteria, if there is any bacteria present, for example, this is the WBC. You have the bacteria that is present. They come inside. Formation of a vesicle is going to take place. Once this vesicle comes inside, you still have identification markers over here present on it, because this is also plasma membrane, right? Like I told you, identification of non-self. Because of this, lysosome comes, recognizes this, fusion with of the lysosome in the vesicle, digestion of the by different, I told you, no, lysosome has digestive enzymes. So whatever bacteria is present inside this is going to be digested by the digestive enzymes or by the lysosomal enzymes. And all the digested solutes, if at all there is anything useful in it, it will be released, or else it will be coming out again, forming through exocytosis. So expulsion of the molecules from the cells. So it can be from the peripheral proteins, ECM, extracellular matrix, or it will be released into the extracellular medium. This is one thing which is there. Let me see if there are any more slides that I have to share with you. We have already seen this. I'm just going through quickly so you will know active transport. I'm not going to talk again because I've just finished these different kinds of ECF, plasma membrane, endocytosis, phagocytosis, receptor-mediated endocytosis. This is where what will happen is there is a receptor that is present, and this is a clathrin-coated pit, as it is known as, because you have the clathrin molecule present, and then it is a coated vessel. Because of this vesicle, what is going to happen is lysosome is going to recognize and come fusion with the endosome, and then degradation or of that particular endosome. What is the cell membrane potential? We have negative voltage inside, positive voltage outside. So minus 40 outside, ECF side, ICF side minus 80. So why is this necessary? For maintenance of the cell integrity and the cell, and used for transmitting signals. So you have a resting membrane potential as well. We will come to this later. You have the Nernst equation also. I don't think this is all there in your syllabus, so I'm just not going into it. Right. We have seen the cell membrane, right? All of us, just a second. We have gone through osmosis. Cell shape and type also, like I told you, it's all different kinds of, I told you, I spoke about the tonicity. Then we have spoken about the carrier mediated transport. Can any one of you actually text me if there's anything left in this particular topic right now, or what else has to be covered as such? On 12, 150. The cytoskeleton, we have just seen the cell components also. The different types of microtubules that are present. Protofilaments, this is the dynein arms. This is the tubulin that is present. Organelles also we have seen different kinds of organelles that are present. Those with membrane, those without membrane, nucleus, mitochondria. Those without a membrane are the ribosomes, centrioles, basal bodies that are present. We have seen about the nucleus, the nuclear envelope, nucleoplasm, nucleoli. So in your microscope, if you see basically, you can use your scanning electron microscope and the transmission electron microscope to actually see these. You're going to get, what do you call it? This is the kind of structure, and this is how you can see inside your scanning electron microscope and the transmission electron microscope. Endoplasmic reticulum with cisternae, we have seen this also. The ribosomes that are present, the Golgi complex, the different kinds of vesicles. What are lysosomes? There are different kinds of functions. What are peroxisomes and the function of it, basically to oxidize the molecules. Proteosomes, okay. We'll have to see that, they're basic proteins. A proteasome consists of the, unfolded protein as such. So this is the target protein. You have two tags that are present just for identification. Mitochondria, double membrane, intermembrane space is also there. Perimitochondrial space as it is known as. Powerhouse of the cell. Centrioles, short cylindrical assembly of the microtubules as such. Centrosome, this is there in the cilia and the flagella. Okay, inclusions are your glycogen granules, pigments, fat droplets, whatever they are like the storage organelles. Foreign bodies, different types. So they're not going to be enclosed in any kind of membrane as such, not essential for cell survival. So this is all about your first unit. I hope this is okay with you. Like if you have understood, or if you have not understood, you're always free to ask doubts. Tomorrow I will be talking about unit 2, that is methods in the cell biology. If there are any doubts, or if you want me to share these slides with you, then you just like I told you, I will share it, or I can share it to any one of you. Just you can message me, then you can share it with your classmates. Or if your group is there, I will share it in the group. So you can always use it for your reference. All right then. Uh, those who, if you want to be added to the common group, I think you'll have to ask or request Kishore sir. Kishore sir's number is over here. Somebody had, wait, wait, let me just send a message to everyone. Send message to everyone, just a second. Kishore sir's number is over here, in fact. Add reaction. See, I just gave a thumbs up. See, you can actually see that is Kishore sir's number. So you can like request him. He will add you because I think he's the group administrator, admin. If that is all, then I will take my leave from the class. Tomorrow, same time, 11:45. It will be unit 2. If there are
Okay,
any doubts, please put it in the group so that the rest of the students can also know what the doubt is. And then if I will try to clarify it whenever the next class is there, or in the group itself, I will send the reference links. I hope
okay.
as of now this is clear, right? All right. Yeah.
అదే పుల్ల తీసుకొని వచ్చేయండి మేడం.
Yeah.
Hello, ma'am.
Yes.
Hello.
Yes, please tell me.
Will you send the reference link tomorrow, ma'am?
Sure. As soon as I get added to the group, I'll try to send. If that is
Okay,
okay
ma'am.
with you. Okay?
Okay, ma'am.
Yeah.
Thank you,
Thank you
ma'am.
so much. Yeah. You all of you can leave for because I think the time is up. Okay? Thank you so much.
Thank you. Okay, ma'am.
Yeah.
Thank you, ma'am. Thank you.
Thank you.
Thank you. Thank you.